DNA. These three letters hold the key to life itself. They represent a molecule so profound that it dictates the traits of every living organism on Earth. From the smallest bacterium to the largest whale, DNA serves as the blueprint of life. DNA stands for deoxyribonucleic acid. It is a long molecule tightly coiled into a structure called a chromosome. Each chromosome is found within the nucleus of a cell, the control center of all living things. In 1953, James Watson and Francis Crick, with Rosalind Franklin's X-ray diffraction images, proposed the double helix structure of DNA. Their groundbreaking work opened up a whole new world of understanding about how life works at its most fundamental level. The understanding of DNA has far-reaching implications, transforming medicine, agriculture, and forensic science. Imagine a ladder twisted into a spiral. That is the essence of DNA structure, a double helix. This elegant structure stores vast genetic information. The strands are made of sugar and phosphate molecules. Attached are 4 bases: adenine, thymine, guanine, and cytosine. These bases spell out genetic instructions. The strands are held by hydrogen bonds. Adenine pairs with thymine, guanine with cytosine. The double helix provides stability and protection. The 4 nitrogenous bases, adenine, thymine, guanine, and cytosine, are the building blocks of the genetic code. These bases pair up to form the rungs of the DNA ladder. Adenine pairs with thymine, and guanine pairs with cytosine. The sequence of these bases determines genetic information. Like letters forming words, DNA bases create life's diversity. Groups of 3 bases called codons code for amino acids. Amino acids build proteins essential for cell functions. The genetic code is universal across all life. This universality shows our shared ancestry. DNA replication is one of the most fundamental processes in biology. It is the process by which a DNA molecule makes a copy of itself, ensuring that genetic information is passed on faithfully from one generation to the next. The process begins with the unwinding of the DNA double helix, like a zipper unzipping. Each strand of the original DNA molecule serves as a template for the synthesis of a new complementary strand. Enzymes called DNA polymerases move along the separated strands, adding nucleotides to the growing new strands, following the rules of base pairing. The end result is 2 identical DNA molecules, each consisting of 1 original strand and 1 newly synthesized strand. This is called semi-conservative replication, as each new DNA molecule conserves 1/2 of the original molecule. DNA replication is a highly accurate process, with only about 1 error occurring for every billion nucleotides copied. This process ensures the continuity of life, preserving the blueprint of life for billions of years. Genes are the fundamental units of heredity. They are segments of DNA that contain the instructions for building and maintaining an organism. Each gene codes for a specific protein or functional RNA molecule, which carries out a specific task in the cell. Genes are like the blueprints for individual parts of a cell or organism. Humans have 23 pairs of chromosomes, 1 set inherited from each parent. This means that we inherit 2 copies of most genes, 1 from our mother and 1 from our father. These 2 copies may be identical, or they may be slightly different, resulting in different versions of a gene called alleles. Alleles are what give rise to variations in traits among individuals. For example, the gene for eye color has different alleles that code for different eye colors. Understanding how genes work is essential for understanding the complexities of life and for developing new treatments for genetic disorders. Imagine a language where every word is only 3 letters long. That's essentially the genetic code, the language of DNA. This code is written in the sequence of nitrogenous bases, adenine, thymine, guanine, and cytosine along a DNA strand. Each 3-base sequence, called a codon, corresponds to a specific amino acid. There are 20 different amino acids that are used to build proteins, the workhorses of the cell. The sequence of codons in a gene determines the sequence of amino acids in a protein, which in turn determines the protein's shape and function. The genetic code is read by ribosomes, the protein synthesis machinery of the cell. Ribosomes read the codons along a messenger RNA or mRNA molecule, which is a copy of the DNA sequence of a gene. Each codon is recognized by a specific transfer RNA or tRNA molecule, which carries the corresponding amino acid. The ribosome links the amino acids together in the order specified by the mRNA, forming a polypeptide chain that folds into a functional protein. Mutations are changes in the DNA sequence. They can be spontaneous or caused by environmental factors like radiation or certain chemicals. Mutations are the ultimate source of genetic variation, providing the raw material for evolution. Some mutations can lead to the development of new traits that give an organism a survival advantage in a particular environment. These beneficial mutations can then be passed on to offspring, leading to the evolution of new species over time. However, most mutations are either neutral, having no effect on the organism, or harmful, leading to disease or dysfunction. Harmful mutations can disrupt the function of genes, leading to the production of non-functional proteins or altered gene expression patterns. Mutations are a double-edged sword. They are essential for evolution, providing the variation that natural selection acts upon. Understanding the causes and consequences of mutations is crucial for understanding the evolution of life and for developing strategies to prevent and treat genetic disorders. The discovery of DNA has revolutionized medicine, opening up new frontiers in disease diagnosis, treatment, and prevention. DNA technology is transforming healthcare, offering unprecedented opportunities to understand, diagnose, and treat diseases at the molecular level. One of the most significant applications of DNA technology in medicine is genetic testing. Genetic tests analyze an individual's DNA to identify mutations or variations that may increase their risk of developing certain diseases. This information can be used for early detection, personalized treatment plans, and even preventative measures. Gene therapy involves introducing, replacing, or inactivating genes to treat or prevent disease. While still in its early stages, gene therapy holds immense potential for treating a wide range of genetic disorders. DNA technology is also playing a crucial role in drug development. By understanding the genetic basis of diseases, scientists can develop more targeted and effective drugs. DNA profiling, also known as DNA fingerprinting, has revolutionized forensic science. It is like a genetic fingerprint, unique to each individual, except for identical twins. DNA profiling relies on the fact that certain regions of our DNA contain short, repeating sequences called short tandem repeats, or STRs. The number of repeats at these STR loci varies greatly among individuals, making it possible to distinguish between individuals based on their DNA profiles. Forensic scientists collect DNA evidence from crime scenes such as blood, semen, saliva, hair, or skin cells. They then extract the DNA from these samples and amplify specific STR regions using a technique called polymerase chain reaction, or PCR. DNA profiles from crime scene evidence can be compared to DNA profiles from suspects or to DNA databases to identify potential matches. DNA evidence has been instrumental in solving countless crimes, bringing justice to victims and their families. DNA technology is playing an increasingly important role in agriculture. Genetic engineering is revolutionizing agriculture by allowing scientists to introduce desirable traits into crops and livestock. Genetically modified, or GM, crops have been engineered to resist herbicides, insects, or viruses, reducing the need for pesticides and increasing crop yields. Other GM crops have been developed to tolerate drought conditions or to have enhanced nutritional value, such as golden rice. DNA technology is also used in livestock breeding to select for desirable traits, such as increased milk production, leaner meat, or disease resistance. Marker-assisted selection, or MS, uses DNA markers to identify animals with desirable genes, allowing breeders to make more informed breeding decisions. While GM crops and livestock have the potential to address global food security challenges, they have also generated controversy. Concerns have been raised about their potential environmental impact, the safety of consuming GM foods, and the control of GM technology by large corporations. The journey into the world of DNA is far from over. As we delve deeper into the intricacies of this remarkable molecule, new discoveries and applications continue to emerge. One exciting area of research is synthetic biology, which involves designing and constructing new biological systems, including artificial DNA and organisms with novel functions. Synthetic biology holds immense potential for applications in medicine, energy production, environmental remediation, and even space exploration. Another promising area is DNA computing, which uses DNA molecules as a computational medium. DNA computers have the potential to perform massively parallel computations, solving complex problems that are intractable for traditional silicon-based computers. The future of DNA research is filled with both promise and uncertainty. It is essential that we proceed with caution, carefully considering the ethical implications of our actions and ensuring that these powerful technologies are used for the benefit of humanity and the planet.